Hey everyone, welcome back to Windows Seed Studies. I hope you all are doing well. So, we started discussing immunology recently, where we learned about what is immunity, what are the different types of immune response that can be evoked by an individual. Then we learned about the hematopoiesis, the process where from the hematopoietic stem cells the mature blood cells are formed. So, hematopoietic stem cells they differentiate into different progenitor cells. The progenitor cells are the precursors for certain cell lineages. For example, the erythroid progenitor is the precursor for erythrocytes, means the RBCs, while the granulocyte monocyte progenitor is the precursor for the neutrophils as well as the monocytes. So we learned about this process in detail. The hematopoiesis means the formation of blood, maturation of the blood cells. So we learned that the hematopoietic stem cells they are very rare in the bone marrow. So they are the resident cells of the bone marrow, and they form the progenitor cells. And those cells, the precursor cells, uh, uh, they are transported to the blood, and they, at that in the blood, those precursor cells give rise to the mature blood cells. So mature blood cells they are found in the peripheral tissue. All of the mature cells, whether it's B cell, T cell, they are found in the peripheral tissue, and the hematopoietic stem cells they are found in the bone marrow. So we learned about the hematopoiesis in detail. Then we started discussing about the cells of immune system one by one. So we learned that the leukocytes, the WBCs, depending upon whether their cytoplasm has granules or not, we have divided them into granulocytes and agranulocytes. Where the granulocytes are the cells, the leukocytes, which have granules in their cytoplasm, and that is why they have a different morphology. We can visualize them under the microscope. So they are the granular ones, and the cells which do not have granules in their cytoplasm, which have a clear cytoplasm, are the agranulocytes. So we learned about granulocytes one by one. So we discussed about neutrophils, then we discussed about eosinophils and basophils. Today we are going to discuss about mast cells. So granulocytes, okay, so they are divided into neutrophils, eosinophils, basophils, and mast cells. Okay, so the difference is that neutrophil, eosinophil, and basophil, all those three cells, all three types of cells, they are blood cells. Okay, so they are found in blood. While the mast cells, these are the tissue resident cells. So they are means they are found in tissues, okay, and not in the blood. So the mast cells are they are also of myeloid origin, just like the neutrophils, eosinophils, and basophils, and they also have cytoplasmic granule. So the mast cells they can be stained, okay, mast cells they can be stained by the purple dyes, and that is why that gives them a purple color. Purple dyes such as the toluidine blue. Okay, so they can be stained by those purple dyes, and the mast cells they have large granules in their cytoplasm, and they have round nucleus. So the nucleus of mast cell doesn't have multiple lobes, such as neutrophils, or bilobes, such as eosinophils or basophils, right? So they only have a simple round nucleus, just like any other cell, and they have large granules. So they look somewhat like this. So let's assume that this is the mast cell. So in this mast cell, there will be a simple round nucleus, okay, and there will be multiple granules, the large granules. So this is how a mast cell looks like under the microscope. Okay, so this mast cell, this resembles to the basophils in the terms that uh, it also has the Fc epsilon R1 receptor. So if you don't know about basophils, please refer to the last lecture. I have discussed about basophils, their structure, the granules composition, and the roles of the basophils in detail. So just like basophils, the mast cells also express Fc epsilon R1 receptor on their surfaces. So Fc epsilon R1 is the surface marker for basophils as well as mast cell. And we discussed that though this Fc epsilon R1, this is the receptor for IgE antibodies. So this is the receptor for IgE antibody. So IgE antibody can go and bind to this mast cell. So the binding of IgE and Fc epsilon R1 receptor, this is going to activate the mast cell, and that activation will result into granulation. Okay, so once IgE antibody binds to its receptor over the mast cell, this is going to result into activation of mast cell, and that will result into degranulation. Okay, so the degranulation means the release of granules in the extracellular environment, and that is why the whatever granules have, that will be spilled in the extracellular environment. So this is same for basophils as well as mast cell. So we are going to talk about this activation process in detail, but before talking about this activation process, let's first discuss about the what does granules have. So this granules, what do they have? So the granular composition, as I told you, that the surface marker, they are present in basophils as well as the mast cell. In the same way, mast cell resembles to the basophils in the granular composition. So of the basophils, they have histamine in their granules. In the same way, mast cells, they also contain histamine, okay, in their granules. So there are a few, there are a few chemicals such as histamine and heparin, which are pre-formed in the granules. So these are known as pre-formed granular components, which means they are already present in the granules. And then there are other chemicals such as other substances such as lipid mediators, which are synthesized after activation. Okay, so histamine, heparin, and lipid mediators are present there in the granules. Okay, so once the granular cell, once the mast cell gets activated, it is going to result into degranulation of mast cell, and that when we investigate the components, whatever components are being released due to the degranulation, we can find histamine, heparin, and lipid mediators. But the difference is that histamine and heparin, both of them, they are preformed, means the mast cell doesn't need activation for having them in the granules. Okay, so they are already present in the granules of the mast cell, even if the mast cell is not activated. Okay, so once IgE antibody comes, it binds to the receptor and results into receptor clustering, that will result into activation of mast cell. And now once mast cell is activated, it is going to synthesize lipid mediators. Okay, so lipid mediators, they are synthesized, so they are not present in the in the granules before, so they are synthesized after activation. Okay, so once the mast cell gets activated, it synthesizes lipid mediators. Okay, so the lipid mediators that are synthesized by mast cells are prostaglandins. We discussed about prostaglandins in the neutrophil lecture, right? So this is vasodilator, and it also secretes leukotrienes. Okay, the histamine, as discussed in the last class, in the basophils class, histamine is a vasoactive molecule. So this is going to control the vascular permeability. So this results into vasodilation. This results into increased vascular permeability. So that why does permeability is increased? Because if the permeability of the blood vessel increases, now the diapedesis or the extravasation of the immune cells from the blood vessels to the interstitial tissue, that becomes easier, and that is why we need certain mediators that are going to increase the vascular permeability, and one of them is the histamine. So this also, alongside the vasodilation, it also causes bronchoconstriction. So I already discussed why bronchoconstriction is important. Well, bronchoconstriction is important because it is going to result into narrowing of the airways. So this narrowing of airways will, it's a kind of protective mechanism, and more than I could say that it is an exaggerated protective mechanism that our body follows to ensure that the pathogen entry is limited. If the physical space, if the width of the airways is decreased, it is narrowed down, so it will result into less space for entry of the pathogen. So bronchoconstriction or bronchospasm is the way of our body to ensure that the pathogen entry is limited. So it limits pathogen entry. Okay, so this is a kind of protective mechanism, an exaggerated one to ensure that the entry of pathogen is limited. So histamine, this is the job of histamine. Then heparin is an anticoagulant. So this is going to result into prevention of coagulation of blood, prevention of blood clotting. So this helps a lot in the smooth immune responses, so that the blood flow is maintained in the inflamed tissue, and the immune cells are able to easily invade that inflamed tissue. Then the lipid mediators, the prostaglandins, they are also vasodilators, so they are going to increase the vascular permeability. So prostaglandins, they are also vasodilators, and they also result into bronchospasm. Okay, whereas the leukotrienes, they also has effect over the vascular permeability. Obviously, they are going to increase the vascular permeability, but besides that, they also result into mucus secretion. Okay, so these are the different components that are found in the granules, different components of the granules of the mast cell: histamine, heparin, and lipid mediators. Okay. So let's now learn about how the activation of mast cell occurs. Okay, so the activation of mast cell, it is the result of the type 1 hypersensitivity. Well, there are different types of hypersensitivity: type 1, type 2, type 3, type 4. We are not going to discuss it here. We will have a separate lecture on it. But for introduction, type 1 hypersensitivity is the IgE antibody mediated hypersensitivity. The hypersensitivity is it is an exaggerated immune response, a bit of exaggerated immune response due to allergens, due to antigens that are entering in the body. So depending upon the type of antibody is mediating this hypersensitivity, the immune responses, they will be exaggerated, but the type of immune responses, type of those exaggerated immune responses will be different, and that is why the hypersensitivity is grouped into different types: type 1, type 2, type 3, and type 4, so on. So these different types have their own characteristics. So the type 1 hypersensitivity reaction is, it results into the activation of mast cells as well as basophils. Means this type 1 hypersensitivity is, this reaction is, is the one where the IgE antibody is going to mediate the whole reaction. So IgE antibody activates the receptors over the mast cells and basophils. So that is why, if the question arises that how the mast cells or basophils get activated, so they get activated due to IgE antibodies, and this IgE antibody activation, this is actually the type 1 hypersensitivity. Okay, so let's learn about the activation of mast cell, or you could say basophils, because both of the cells, both of the immune cells, they get activated by the same mechanism. So I am going to discuss it here. So as I already told you, for the activation of mast cell or basophils, the important thing that is required is the receptor clustering. Okay, so let's assume that this is the mast cell. Okay, so over this mast cell there exist the receptors. So these are the receptors that are present over the mast cell basophils. These are Fc epsilon R1 receptors. Okay, so those receptors, when they get clustered, you remember when we started discussing about signaling, when we discussed about receptor tyrosine kinases, for activation of the those receptor tyrosine kinases, dimerization was very important. So when those receptors got dimerized, they activate the intracellular signaling pathway. In somewhat similar way, the activation of mast cell and basophils, it requires the receptor clustering. Means all of those receptors that are present over the cell surface, over the surface of mast cell and basophils, they need to get clustered in some place, and this clustering is going to result into the intracellular signaling. Okay, so once this receptors get clustered, okay, they will induce the intracellular signaling cascade. Okay, so for the receptor clustering, so we can say that activation of mast cell and basophil is due to the receptor clustering. Okay, so which receptor clustering is required? The Fc epsilon R1 receptor clustering is required. Okay, so how will those those floating receptors are going to get clustered in one group? So they will get clustered in certain area if something comes and binds to those extracellular domains. And as we discussed before, to those receptor, to those Fc epsilon R1 receptors, the IgE antibody binds, right? So IgE binding is going to cluster those receptor molecules, but not just antibody is going to mediate this clustering. There are multiple ways by which this is controlled. So what happens is that the IgE antibody is going to go and bind this receptor. So let's say that this is the antibody. Okay, now it is bound to the receptors. Okay, so even though antibodies are bound to those receptors, so let's call this IgE receptor complex. Okay, so once this IgE receptor complex is formed, this again is not ensuring the it is not ensuring the clustering. So what ensures the clustering? The property that the antibody possesses, a property that is referred as cross-linking. So cross-linking of this antibody is going to result into the receptor clustering. So first let's learn about what is cross-linking, because I haven't discussed about antibodies yet, so you might not know about it, and if you know, that's a good thing. So if you don't know about anything about antibodies, don't worry about it. We are going to discuss about their structure, their function, their formation, okay, in detail in some separate video. So for now, what you need to know is that antibody, these are the these are the glycoproteins. Okay, so these are molecules which possess a property known as cross-linking. Means they have a binding site not just for receptors, but they are multivalent. Means they have multiple binding sites. They can bind to other antibody. They can bind to either some allergen, some antigen, or there exist bivalent antibodies. Means this Y-shaped antibody can bind to two antigens, okay, bivalent antibodies. So this means that antibodies could be multivalent. So they can bind they have multiple binding sites. One of them binds to the receptor, forming IgE receptor complex, and other binding site is going to interact with the antibody, the IgE, which is already bound to receptor. And this phenomena is known as cross-linking. Okay, let me explain it again. So what will happen? IgE antibody will come and it is going to bind to the Fce epsilon R1 receptor. But as this IgE antibody it is going to cross link, means it is going to interact with the antibody which is present, which is already bound to some other receptor. So the interaction between those two antibodies is known as cross linking, and that is going to bring those two receptors together, and the multiple receptors will be clustered in some area, in a particular area, resulting into the activation of the mast cells or the basophils. So what will so to the receptors IgE will bind. So by the cross linking, okay, by cross linking the IgE antibody, we can activate the mast cells. Okay, so this cross linking of IgE antibody, this could be either the antibodies themselves they are cross linking, or this could be due to multiple reasons. First, it could be chemical cross linkage. Okay, so IgE antibody, if it binds to the receptor, let's say that there is some chemical mediator, some chemical which has binding capacity, which is kind of divalent. Okay, so one arm of this chemical can bind to one IgA antibody, and other arm can bind to other antibody. Okay, so this is going to cluster the receptors together. This is going to bring those IgE bound, IgE receptor complexes together, resulting into the activation. So chemical cross-linkage can be one way to ensure the receptor clustering. Okay, the other way to ensure the receptor clustering is, the multivalent allergen. Okay, some allergen itself has the binding capacity. It is already multivalent, and it is going to induce the cross linkage. So we can call it the allergen induced cross linkage of antibodies. Okay, the certain allergen can come, and this allergen has binding capacity. Okay, so it is multivalent, it can bind to multiple IgE antibodies, and it will result into receptor clustering. Okay, so this is how the cross linkage of the antibodies will be seen, and that is going to result into the activation of mast cell. The other way of ensuring that those antibodies are coming together, they are clustering together, is that we introduce other antibody, which is anti-IgE. Okay, so this is IgE. Okay, IgE antibody. So this binds to the receptor Fc epsilon R1. So we can call IgE antibody as anti-Fc epsilon R1 antibody. Okay. So if a certain antibody comes and binds to IgE receptor, means let's say that there's an antibody and it has capacity, to bind to this IgE antibody. So this antibody, this particular antibody will be referred as anti-IgE antibody. Okay, because it is binding to IgE. So such anti-IgE antibodies, they can also bring together, if they are bivalent, means they can bind, or they are multivalent. So those anti-IgE antibody, they will result into binding to the multiple IgE, bringing the receptor closer. So binding of anti-IgE antibodies will also result into the receptor clustering. So the cross linkage of the IgE antibody will be also, that can be also possible due to anti-IgE antibodies. So there are different types of anti-IgE antibodies, one which can bind to variable region, while other can bind to constant region. So depending upon that, they are different, such as anti-isotype antibody and anti-idiotype antibody. Okay, so well, if you don't know about those types, fret not, we'll discuss about them later. So this is how the anti-IgE antibody can also result into the cross linkage of the IgE antibodies. Okay, so other way by to ensure that those receptors get clustered is to, if is to have a molecule which can directly bind to those receptors. So now those IgE receptors, they are bound to the Fc epsilon receptors one by one. So let's assume that there's some molecule, there's exists some molecules which has a capacity to bind to multiple receptors itself. So we won't need IgE antibody, right? So there could exist some antibodies other than IgE, which has capacity to bind to multiple receptors. Okay, so IgE antibody can only bind to one receptor, right? So if there exists some certain antibodies, and those antibodies can they can result into clustering of the receptors. So there could exist some anti-receptor antibody which is going to result into clustering of the receptors, and that will also activate the that will also activate the receptor. Well, I don't, in this schematic, I made a mistake. When we draw the antibodies binding to the receptor, I should have drawn it something like this. Okay, so Ig antibody will come and bind to the receptor in this manner. Okay, so there could be some other antibody which whose two arms can bind to multiple receptors, two receptors, and that can result into clustering of the receptors, resulting into the activation of the mast cell. Okay, so these are the different mechanisms which will ensure the cross-linking of the Ig. So these three mechanisms, okay, they are going to ensure the cross-linking of IgE antibody, and this is other way of clustering the receptors. Okay, but there's also other mechanism to ensure the activation of mast cell, and that is calcium influx. Okay, so let's say that calcium ions, they are getting they are they are transported inside the cell, then mast cell can also get activated, and that can be ensured by the ionophores. So let's say that there exists ionophores over the mast cells, which can directly induce the calcium influx. Then we can also activate the mast cell. So these are the different ways to activate mast cell or basophils. Okay, so in short, in one sentence, if I have to define the activation of mast cell, we could say that the mast cells or basophils, they get activated due to the Fc epsilon R1 receptor clustering, which is mediated by IgE antibody. Or in other words, we can say that type 1 hypersensitivity reactions are going to result into activation of mast cells. So activation of mast cells or basophils, it is IgE mediated. So with this, I'm done with the mast cells. Okay, so in next lecture, we are going to learn about the other types of immune cells, such as dendritic cells, macrophages. Okay, so with the mast cells, we are done with the granulocytes. We have completed all different types of granulocytes. Then we'll learn about dendritic cells first, then the mast cells, the macrophages, and then we'll start discussing about lymphocytes, B lymphocyte, T lymphocyte, as well as the natural killer cells. Both of them belong to the lymphoid lineage. So till then, so I'll catch you in next lecture. Till then, see you. Bye bye.